Thank you. So my name is Teresa Rummel, and I'm a PhD student at the University Regensburg in Germany. And today I want to talk to you about how we can make use of single-cell metabolic labeling data in order to learn about transcriptional bursting. So first off, transcription is an inherently stochastic process. So it depends on the availability of molecules at the right place and time in order for active transcription to happen. So if all of those things align and we have a promoter switch from the inactive to the active state, usually multiple mRNA polymerases transcribe the loci at once, generating those spurts of mRNA or bursts. Afterwards, it switches into a deactive state again, and there, of course, we have degradation. Of course, we can also look at this process at like the number of mRNA molecules over time, and what we see there is this pattern, this wavy pattern that gives us the chance to look at bursting with certain parameters. So the parameters that we usually use to characterize this is how often do those bursts occur, or the burst frequency, and how many mRNA molecules are usually made during an active period, which would be the burst size. And of course, also the how fast the molecules are degraded again, so the decay rate are important. Already in steady state, those parameters are quite different for different groups of genes. But more interestingly, these parameters also play a big role in regulation. So here I have a little toy example where we look at 10 cells and 5 of them express one transcript with one copy. So now we get a virus infection that will induce this transcript. And this could go in two ways. One would be the upregulation, where we have double the expression of this transcript in the same cell. The other would be the mode of on-regulation, where we now have expression in all cells, but still the same level as before. And of course, those are two extremes. There may also be intermediates between those. In bulk, we would not see the differences here, but in single cell, we actually can see that this exists, and we already saw this in some real-world data. So what we have here is two different conditions. One is MOC, and the other is MCMV infection. And this would be an example for upregulation. So we have here more mRNAs per burst, and almost in the same amount of cells. And I also have one example for the on-regulation. So here we see more swift to the right, whereas here it's more towards the up. And so we have the same level of expression, but now in almost all cells, whereas before it was only in about 60% of the cells. So we really wanted to know more about this, and previously there already has been quite some approaches to infer bursting with normal single-cell RNA-seq data. So let's say we have one gene that is expressed at a certain level in a bunch of cells, and we can also look at this as the distribution of the total RNA. And of course, there's also a mathematical model for this. Usually it's the two-state model that basically just models the promoter, which can be either in the on or off state, and it can switch between those states with the certain rates. In the on state, transcription will take place with the synthesis rate, and if we have mRNA molecules, they will be degraded with the certain half-lives. For this model, a closed form mathematical solution exists that we can basically just calculate for any types of parameters and look at the distributions that we get from that. And then we can basically just do a maximum likelihood estimation, use our real-world data, and look which parameters fit the best. So far, this has a few issues. One is that, I just solved the mathematical model for one set of parameters, and this is the distribution that we get. But with normal single-cell RNA-seq data, we have no kinetic information, and we do not really have information about the half-lives. So if we halve the half-life and double everything else, we could get exactly the same curve. So this approach does not take into account that we have gene-specific mRNA degradation rates. Also, single-cell RNA-seq data is noisy, and if I leave the half-life the same and just switch the parameters a bit, I get a very, very similar curve. So it's hard to tell if the parameters are really that perfect. And single-cell RNA-seq data can only capture like one cell once, so we don't get the time resolution. So it only will work in steady state. So we cannot look at all this whole regulation thing that I just showed you. So this is why we use it with a metabolic labeling single-cell RNA-seq data. And our lab usually uses SLAM-seq data, which also uses the nucleoside analog FORSU that we already heard about. And the cells get labeled with FORSU, and this then gets incorporated into newly synthesized RNA instead of uridine. In SLAM-seq, then this FORSU gets converted into a cytosine in newly labeled RNA, whereas uridine, of course, then gets into a thymidine. And then after RNA sequencing, we have those T2C mismatches in our reads. And based on those mismatches, so if the read has mismatches, we knew that it was newly made, whereas if it's no mismatches, it's null read. Unfortunately, this whole process is also not perfect, and for SU incorporation only happens at a quite low percentage of U sites. So with short read data, we have the issue that we will end up with quite a bit of new reads that may not contain a single FORSU, and therefore in the end will also not contain any mismatch. Our lab previously came up with a solution for this, and this is by using a Bayesian hierarchical mixture model. We estimate the new to total ratio for each gene, and so we basically get an information about how many of the RNAs were new and old per cell and per gene. And additional to this point estimate, we also get the posterior distribution, so we know how good the estimate is. And we can also take the posterior to downstream methods like the bursting estimation. So now that we have the single-cell SLAM-seq, we basically do not end up with one distribution, like I showed you before with the normal single-cell RNA-seq, but we end up with two distributions. We have a distribution for old RNA and a distribution for new RNA per gene. With that, we went back to the original models for transcription, and notably this is the steady-state model. And we then started to adapt it until we were at a point where we got two different mathematical models, one for old and one for new RNA, that we can solve at any time point t, such that it will also work under non-steady state conditions, and we are able to look at regulation. Unfortunately for those, we don't have a nice closed form solution, so this was computationally quite expensive. And that's why we also came up with an approximation for this using the method of moments. So this is now all in place and lets us compute like also all of the distributions for new and old RNA in a very effective manner for SLAM-seq data. And what we do with this is basically just a joint maximum likelihood estimation with just more information than before. And using this, we can estimate the half-life, the burst sizes, and the burst frequencies for each gene in single-cell SLAM-seq data. First, of course, we went ahead and tested it with a lot of simulated data, but for the sake of time, I'm not going to show you this today, so you have to believe me that it works. And I really wanted to show you a real-world application. And for this, we went to one gene that is very interesting, also the regulation of downstream targets. So we looked at MYC. So MYC is a gene that can bind to RNA and influences the expression of a lot of downstream genes. And it's a very important gene because it controls the cell proliferation, growth, and apoptosis, and it plays a major role in a lot of cancers. And for us, it was also very interesting because there has also already been a publication where they did live cell imaging using MYC. And they looked actually on how it influences it. And what they found is that MYC regulates downstream targets by increasing the on time of the genes, which would correspond to a higher burst size. So with all of this in mind, we teamed up with Elmar Wolf in Kiel, and he's working a lot with MYC, and they have a very cool system in place. So what we did is we took two different cell lines. One is MAML63. This cell line has low endogenous MYC levels. And the other cell line is A375, which has high endogenous MYC levels. For both, they have an auxin-inducible decron system in place. And based on this, then MYC was depleted from the cells, and then we did single-cell SLAM-seq. And first, I just did normal gene expression analysis and looked at the UMAP of total RNA. And what we can see here already is that the A375 in the mammal cells separate nicely. And in the mammal cells, all of the conditions look quite heterogeneous, whereas in the A375 cells, it already seems that there's a bigger difference between DMSO and auxin-treated cells. Then I went ahead and looked at the new RNA and calculated a UMAP for that. And so this is the cell line with the low endogenous MYC levels, and here in the UMAP, we don't see a clustering based on the treated or not treated cells. Whereas in the A375 cells, those nicely separated, indicating that we see a strong effect if we have higher endogenous MYC levels. I also calculated a module score for some previously published MYC signature genes, and this nicely showed the same thing. So in the DMSO cells, we have way more expression of the MYC signature genes, whereas in the auxin-treated cells, where we have the MYC depletion, we have lower levels of those genes. Then I did a pseudobulk analysis, just doing log-fold changes between the auxin and DMSO conditions and looking which genes were regulated. And here we basically see that also in the mammal cells, we see some downregulation of quite some cells. In the A375 cells, it's more extreme. And yeah, so it's a strong effect with the A375 cells because we had their higher MYC levels, and it's a lower effect, but still an effect in the mammal cells. So then I applied my bursting model. First, I looked at all genes, and yeah, I mean, there was a little bit of everything and also a bit of noise. Then I took the mixed signature genes and looked what are the major changes here. And I see that most of those genes are either gene change in frequency or frequency and burst size, so both. And there are also a few cells which have changes in burst size. Then I took all of the genes that had a log-fold change smaller -1 on the previous log-fold change plot, and it's kind of a similar picture. We see a lot of genes that changed in burst frequency or both, and a few genes that showed changes in burst size. So this looks way more heterogeneous than what the paper I showed before suggested. This suggests that there was mainly changes in burst size. And I also want to show you two example genes. So this would be one gene where we had mainly changes in burst frequency, and it looks very similar to what I showed you in the introduction with the regulation. So we see here this off shift, basically. So this is similar to the on shift in the introduction. And in contrary, there's also a downregulated gene where we had seen mainly changes in burst size. And here it's this downregulation. So we see that it's still almost expressed in the same cells, but less. Yes, and with this, I would like to summarize. So we developed a mathematical model that can handle single-cell metabolic labeling data in order to estimate burst frequency sizes and degradation rates. And it works under steady state and non-steady state conditions. And when we applied it to the MYC data set, we see that MYC might have a more integrated role than what we previously known. And yeah, in the future, we want to apply this for multiple data sets and like I already said in the introduction, we also have this posterior distribution from the NTR, and I'm right now also working on including this in my burst size, burst parameters estimation. And with this, I would like to thank my whole group and everyone involved in this project, and of course you, and I'm happy to take questions.
So in single-cell RNA sequencing experiments, we have this dropout phenomenon where in certain cell types, there are genes that we're expecting to see expressed in all cells, and yet a subset of cells there's no expression. And it could be due to a technical artifact in how the experiments are run, or it could be due to this bursting phenomenon. Can we use this technique to try to understand the relative contributions of those two phenomena to this dropout?
I mean, we aim to, but it's hard. Of course, it's always hard to tell if the noise is more biological or more technical. I would say the dropout effect here is I would think that it's mostly to all cells and it's not to a subset of cells. And so the bursting parameters would be just like reduced to a certain degree.